So let's do just a little course mechanics. I just want to remind you about the Moodle page and what's available there. Here are YouTube's, this is a YouTube playlist from last year, which you can use to look ahead. And this is the playlist for this year, which is your source to review material from what we have talked about in class today, or any day. If you need to Zoom, this is the Zoom for classes, and here is the Zoom link that you'll use today for a breakout session, that will set up a breakout where you can work together with each other to teach each other and learn from each other how to do, answers to some of the problems. Brittany and I are co-hosts of this meeting, so we will be available. So if you're in a meeting and if you get stuck, you call for help from a breakout. There's a call for help button. You call for help, and one of us will come and help you work to answer that problem. Those of you who know material, teaching others is the best way to really solidify your knowledge. Those of you who are confused, hearing it from your peers is often the best way to get clarity, because sometimes I use language that isn't the best for you. So I think working together on the problem sets is going to be a very effective way for you to learn the physiology, solving problems together. So here are the PowerPoints from Monday, which now have answers. Right? The links look the same, but the PowerPoints that are available now are different from the ones that were available on Monday because they have the answers to the clickers. I recommend that you don't just look at my answers, that even as you review it, you try to answer the question from the blank one, which precedes the one that has the answers, because solving a problem is more active than just seeing a problem, an answer. And I feel strongly that active learning, writing, speaking, outlining, cartooning, labeling, anything when you're doing something is a much more effective way to learn than reviewing, skimming, reading, listening. Those are passive, and things don't get into your brain as good, as well, as when you're active. If you have a Windows computer, you can download the simulation, but maybe everybody should just use this link, which works. The link in your problem set does not work. So my recommendation for the breakout is the first thing you want to do is open up Moodle, then open up the problem set text, which is embedded in this link, to have that in front of you, and then open up the calculator to have that link in front of you. And then if you're working on problem 2 together, this is your tool. And then here we are today, PowerPoints for today. So we'll work here together for, 45 minutes or so. Then we'll have a 10-minute break while you'll go to Zoom locations. Then we'll Zoom then you'll break out and work on problems for 45 minutes. And then we'll come back here, either in person or on Zoom, to do this class. And this will be a pretty typical Wednesday. So I've been working on lab assignments. I hope I got it right. So check it and look for your name on this. Make sure you're in the section that works for you. If you're not, we need to work it out, maybe on a Zoom call or by email this afternoon. Let's not use class time. So if there's an issue here, let me know this afternoon and we'll work it out. So today we're going to finish GPCRs, talk about the turning off of GPCRs, and then we're going to start to get into some neurophysiology. Cellular neuroanatomy and neurophysiology. My goal is to teach you material so that the problem set is on a foundation of some understanding. Okay, so here's the GS story that you learned about in self-phys, that I asked you to review and write a problem for homework. I'm not going to go through it because you can learn about it from the book, from your homework, from self-phys notes, etc. What I want to point out now, though, is that there are ways that this system, that this cascade, turns off. The ligand, the agonist, can unbind from the receptor. And so whatever was triggered by the binding is untriggered by the unbinding. The G protein, which is active when it is in the GTP bound state, when alpha subunit is bound to GTP, T as in Thomas, it's on, it's active, but alpha subunits have an intrinsic GTP ACE activity. They hydrolyze GTP. So they are activated by GTP, and then they hydrolyze GTP and turn themselves off. So there's an intrinsic turning off process that has kinetics that are slower than the turning on. So it turns on, and then they turn off. And that turn off is intrinsic to the G protein and is accelerated or inhibited by other cellular factors that you can learn about if you're, doing advanced study in G proteins. The point is there's an intrinsic hydrolysis of the GTP back to GDP, which is the off form of the alpha subunit. Adenylyl cyclase turns ATP into cyclic AMP. There is an intrinsic turning off there. Adenylyl cyclase converts ATP into cyclic AMP, but there are constitutively active enzymes in the cell that hydrolyze cyclic AMP to AMP, which is not active. So adenylyl cyclase is turned on by an active G protein to make cyclic AMP. And then phosphodiesterase, which is there sort of in the background, hydrolyzes ATP to AMP. Now you can predict that the kinetics of making cyclic AMP are faster than the kinetics of breaking it down, so that cyclic AMP is present for some period of time, triggering events like kinase cascades. But then the cyclic AMP is hydrolyzed and turned off. Hydrolyzed, and the on switch is turned off. So the turning off is almost as important as the turning on, because if we don't turn off a system and we only turn it on and it stays on forever, that's disease. And that's not this course. This is a physiology course, not a pathology course. Okay, so here's one of the ways that cyclic AMP is turned off by hydrolysis. And you should also remember that anytime there's a kinase, there's almost certainly a phosphatase, again, working in the background to dephosphorylate the things that a kinase turns off. So we have unbinding, GTP hydrolysis, cyclic AMP hydrolysis, dephosphorylation as the possible steps for turning off. Here's the GQ GQ story, which your problem set asks you to outline. So I'm not going to do it because I want you to do it actively by solving the problem. And then there's a question about turning off the GQ pathway. And I want you to work on that. I actually made the question much too easy because I said there are at least 2 ways, but I could make it harder by saying there are at least 4 ways. Mapping your knowledge of the things that turn off GS onto the things that turn off the GQ story. Okay, that's part of your problem set. And you can work on that problem together. Oh, during the breakout, what I want you will have to decide which problem you want to work on. You won't have time to work on all of them. So you and your teammates in your breakout room will prioritize. Okay, let's work. Can we work on questions 5 first? Can we work on question 1 first? Let's move on. I think we're all pretty clear on what we need to do. Let's move on to question 2. So, work together to decide how best to use your time in the breakout. Okay, let's turn our attention to neurophysiology. And we're going to start at the cellular level. Cellular neuroanatomy, neurophysiology. The morphology of neurons is, cellular neuroanatomy, the structure of neurons. The function of neurons is neurophysiology. That's what we're going to do now. So here's a cartoon from your textbook, cartooning the cell body, dendrites, cell body, there's the nucleus. Obviously, all the other things that you learned about in cell-phys are in here. Endoplasmic reticulum, mitochondria, cytoskeleton, endocytosis, exocytosis, metabolism, all of that stuff is going on here, but it's in the background here. Dendrites are where the receptors are. Cell body is where the cell is. All that cell physiology is. The axon is the output pathway. So input, processing, output, and then a synaptic terminal. And so all of these little ends of this dendritic, of this pathway, these are all making synapses onto the next cell in the pathway. So here's a cartoon. Here's a drawing that is more realistic. Here are images taken from microscopes. So these are data of neurons. Dendrites, cell body, axon. Cell body, axon collateral. Incredible dendritic arborization. You can imagine how many synapses there are on this one cell. Tens of thousands. This is a cell in your cerebellum. This is a cell in your hippocampus. We'll talk more about those anatomical structures in a few days. Here's a cell in your cortex, your cerebral cortex. Okay, so it's complicated. Neurons are special because of their complexity. So, in the past, when there was more time to do this, I did this as a game where I passed a laser around. Well, we can't pass a laser around anymore. And I asked you to identify which dot is which name. Remember, naming things is anatomy. And so this is a slide that asks you to name some of the structures of a cartoon nerve cell. So let's do some of it, but we're not going to spend a whole lot of time with it because I want you to do this as homework. So for example, what's this dot right here? That's the nucleolus. We don't even talk about that in cell phase, so forget about it. This thing here, there's the nucleus. That's pretty easy. Dendrites. That must be the soma. Soma is another name for cell body. So when we talk about somatic something, we're talking about something on the cell body. If we talk about dendritic something, we're talking about something on a dendrite. If we talk about axonal something, we're talking about something on the axon. If we talk about terminal something in this context, a terminal something would be something in the terminals of this neuron. It gets a little ambiguous. This arrow, what is that? That's the direction of signal transmission, right? So what are these sort of cloudy things going along this axon? Those are the Schwann cells that myelinate axons and provide insulation. We'll talk about that in a few minutes. So you could say that this is Schwann cell. This game also calls them, internodes. Let's not worry about that. These are Schwann cells. Where the axon is not insulated with the Schwann cell myelin is the node of Ranvier. So you can play this game. I think you should play this game, maybe more than once, to learn the cellular morphology of nerve cells. So, I know you can barely see this. Let's focus over here. So, what makes myelin are Schwann cells and oligodendrocytes. In the periphery, in the peripheral nervous system, Schwann cells. In the central nervous system, oligodendrocytes. The function is the same, to wrap axons with myelin and improve signal propagation. So there's the node of Ranvier. This cloudy gray stuff, which you can now barely see, is the myelin. And you can see here, the green label is labeling voltage-gated sodium channels that we're going to talk about in detail right now. And the pink label is voltage-gated potassium channels that we're going to talk about in a few minutes. So channels highly concentrated at the node of Ranvier and nearby. Myelin insulating the axon. So now I want to transition from cellular anatomy to cellular physiology. And what we're going to talk about is the creation of membrane potentials. In this room, when we talk about potentials on this context, we're talking about voltage, membrane voltage. Voltage is a concept that sometimes is hard to get a grasp on. Voltage is a separation of charge as a function of space. So if I put a positive ion here and a negative ion here, there is a voltage between them, a charge separation, an electric field, for those of you who took physics, creating a voltage. That voltage is an electrical potential. So we're going to talk about membrane potentials. which are voltages. And how this, you heard from self is that the resting membrane potential is negative. We're now going to unpack that a little bit more. So, creation of membrane potentials. Generally, and then we'll get specific. So here's a cartoon of a membrane separating two compartments with a solute on both sides of the compartment. So let's say I gave you this. Let's call this compartment the inside and this compartment the outside. Right? So in a cellular, this is the cytoplasm and this is the extracellular space. Let's say that these dots represent potassium. There's more potassium on the inside than on the outside. Why? Active transport. Sodium potassium ATPase pumped those potassium ions in there. If this membrane is permeable to potassium, potassium will flow from inside to outside, down its concentration gradient. That's a spontaneous reaction, energetically favorable for things to diffuse down their concentration gradients. There's an equation, those of you who are physically oriented, there's an equation that defines the flow due to diffusion. It's a constant, some diffusion constant, and there's the concentration gradient. Concentration differential as a function of space. So that's the concentration gradient, and that's the diffusion constant. And just to make you feel like, okay, yeah, this was complicated stuff, the diffusion constant is a function of lots of things that you don't have to know, do not have to know. But this is Albert Einstein. Albert Einstein, who you all know, have heard of, did work on understanding diffusion. So it's not very often that in a biology class we get to introduce Albert Einstein, and so that's why I do it. So let's now take that knowledge and understand the creation of voltage. So here's the same thing we just were talking about, just to be consistent. We're going to call this the inside and this the outside, just like we did before. This inside has lots of potassium ions. So each of these black dots and of course there is some chloride and other counter ions, anions, negatively charged ions, on the inside. And there is some potassium on the outside. And there's actually quite a lot of chloride on the outside. And if you're interested, I can explain why there is a chloride gradient, but that's part of one of your problems, actually. Not to define it, but just to use it. Okay, so there's the starting point. What created the potassium gradient? Sodium potassium ATPase. Pumped potassium into the cell. The membrane is permeable to potassium and not to any other ion. The resting permeability of this membrane is permeable to potassium, not to sodium or chloride. The channel, the name of the channel that is responsible for the K permeability is we'll just call it the resting potassium channel. They are open at rest. they are undergoing conformational changes. They are opening, closing, opening, closing, closing, opening, opening, you know, they're undergoing the closing and opening conformation, but they're doing that without any stimulus. It's a constitutive process. They are not open all the time, but some of them are open all the time. So, when one of them opens, a potassium ion could flow down its concentration gradient, delivering a positive charge to the outside and leaving behind a net negative charge on the inside. Right? Remember, it's selectively permeable. Only potassium can flow. So another one goes through an open channel, another negative charge left on the inside. Again, negative. Okay? So now you're already seeing that the inside of the cell is negative relative to the outside of the cell. And the convention is always to refer the inside relative to the outside. So when you think about membrane potentials, you want to think about what is the voltage on the inside relative to the outside. Vincent?
Right, so I get that the residual negative charge from potassium going to outside of the membrane is what causes that negative charge inside, but are there not more potassium ions inside the membrane which would have a positive charge?
There are more potassium ions here than here, but this situation is electroneutral.
Oh.
There was more potassium, but there was an equal number of anions. Some of those anions were chloride, and some of those anions were negatively charged proteins or phosphate, which is negatively charged. So out here in bulk solution, it's neutral. The voltage is limited to a very narrow nanometer region around the membrane. But you're right, at around the membrane, there is an imbalance. There is more potassium on the outside than on the inside, within nanometers of the membrane. That's not true in the bulk solution. Okay, so we've created a negative voltage. The voltage is negative. Now, Brittany, I just got a message that our breakout room has closed because it's inactive. So we need, when we go to breakout, we'll have to restart. Help me remember to do that.
Okay.
So now, let's think about this voltage. This voltage is negative on the inside due to the concentration gradient and the selective permeability of the membrane. But these ions out here, this potassium ion, and this one, and one that might be here, they're positively charged. They respond, they can sense, in a way, this negative charge that isn't very far from them, nanometers away from them. If this potassium ion is very close here, what direction is it going to move in response to the electrical potential as opposed to the concentration potential? It's going to move that way. So there is now a voltage flow that's due to some constants. This is a constant. This is the charge. This is the concentration on the source side. And here is the voltage gradient. So we have one equation that defines the flow in response to the concentration gradient. Again, you don't have to know the equations. I'm just trying to make this make sense. A flow in response to the concentration gradient this way, that's easy. And now we have a flow going the other way in response to the voltage gradient. And we've got opposing flows, and so you can imagine already, this is going to set up an equilibrium. For every potassium that flows this way down its concentration gradient, there's going to be a potassium that flows this way down its electrical gradient. And if we solve that equation for the zero flow situation, that's equilibrium. At equilibrium, there's no net flow. Every time there's a potassium moves this way, a potassium moves this way. No net flow. There's flow going on, but no total net flow. We are at equilibrium. If we solve that equation, and it's not we, it's a guy named Nernst, we get an equation. And this equation you're going to be using in the problem set. You're not going to have to dig out your calculator because the online calculator does it for you. But this is what's running in the background for some of the problems. The voltage that is set up in equilibrium by a concentration gradient is defined by this equation. This is the solving of the flow due to concentration and the flow due to voltage. What voltage does it go to get at equilibrium is defined by this equation. This is just a conversion of the equation, turning natural log into log base 10. You're not going to have to use these equations because you're going to have a calculator that will use it for you. So this number, which is in millivolts, don't give me numbers in volts, give me numbers in millivolts, it's expressed in the problem set. That's the equilibrium potential. These are constants. This is the charge of the ion we're talking about. So your problem during the problem set is to use this equation. But I want you to understand it using English words. And so this slide, I worked pretty hard to try to make it make sense using words as opposed to equations. The equilibrium potential for a specific ion, it's always for a specific ion, potassium, or sodium, or chloride, or calcium, is the voltage that that membrane would go to if it were permeable to that ion and only to that ion. That's part of what that would allow you to get to equilibrium. Because the equilibrium potential, there's no net flow of that ion if you are at the equilibrium potential. Channels could be open, but for every time there's a channel moving in one direction, an ion moving in one direction, there's an ion moving in the opposite direction. No net flow. If the membrane potential is above, more positive than the equilibrium potential for that ion, we get outward flow. If the voltage on the inside is above, more positive than the equilibrium potential, that will allow outward flow of the ion. And if the membrane potential is more negative than the Nernst potential, than the equilibrium potential for that ion, that will allow inward flow of the ion. And this is assuming the ion is positively charged. If the ion is negatively charged, things flip, and you might have to figure that out in the problem set. So I want you to think about these things when you're solving the problem. Problem 2, mostly on the problem set. So here are some typical gradients for ions. Don't use these numbers in the problem set, use the number in the problem set, but here are some more typical numbers. High concentration of potassium inside. High concentration of sodium on the outside. High concentration of calcium relative to the inside on the outside. That's a high concentration relative to that. Chloride, higher on the outside than on the inside. You'll use some numbers to solve these problems. And so if you use these concentration gradients to solve the equation, you get equilibrium potentials. And so these are typical values for the equilibrium potentials. Large negative for potassium. Large positive for sodium and calcium. Variable negative for chloride. So now let's turn our attention to what happens if we have a real cell with multiple permeabilities. Remember, the Nernst potential is only correct if the membrane is permeable to one ion. But cells are permeable to multiple ions at the same time. There are resting potassium channels. There are leak sodium channels. So let me blank this slide for a moment. Raise the screen. Go back to this picture. So this is equilibrium for potassium. Now let's add sodium. Okay, so orange. No, let's make green. Green is sodium. High concentration on the outside, low concentration on the inside because of the sodium-potassium ATPase. At rest, there is low, but not zero, sodium permeability. So we have, in addition to the resting potassium channel, we have some leak sodium channels. Okay, so if we have, let's just put one leak sodium channel in here. Which way is sodium going to flow if it responds to its chemical gradient? That way. Down its concentration gradient. Which way is sodium going to flow if it flows down its electrical gradient? Same way. Negative on the inside. Sodium is a cation. It's flowing that way. So there, if we had one leak sodium channel, we're going to get some sodium flow. Sodium is a cation. So it's going to neutralize some of that negative charge and displace this membrane away from equilibrium. It's going to be less negative on the inside than it would be if it was only permeable to potassium. So sodium leak makes the membrane potential depolarized. Now I'm starting to use some of the jargon. Depolarized relative to the equilibrium potential. And that's why the resting membrane potential, which you know from self-phys is around minus 70, minus 65, minus 75, depending on the cell and its permeabilities. The resting membrane potential is not equal to the equilibrium potential for potassium. It's less negative than that, minus 60, because there's a little bit of sodium permeability at rest. There's an equation that, again, you don't have to memorize, you do not even have to use, because it's embedded in the calculator that you're going to use online. But this is the equation. What does this mean in English? The voltage of a membrane is equal to some constant in a function, and the function depends on the gradients, potassium on the outside, potassium on the inside. The potassium gradient, the sodium gradient, the chloride gradient, and in fact the calcium gradient, but that because calcium makes this equation blow up. So we'll just use the monovalent ions. Monovalent cations and monovalent anions. So the membrane potential is a function of the concentration gradients and their relative permeabilities. The permeability to potassium, which is high at rest, so this term dominates, and the permeability to sodium, which is low at rest, and the permeability to chloride, which is variable at rest. So this is the equation, the voltage that a membrane would have given the concentration gradients and the permeabilities. And this is the equation you're going to work with in 5-10 minutes. The language of voltage is, we talk about a resting membrane potential, which is, about -70, but variable depending on the cell. So it's not a number worth memorizing, but it is worth remembering that the resting membrane potential is negative. It's not as negative as the equilibrium potential for potassium. It's not anywhere near the equilibrium potential for sodium, because the permeability to potassium is high and the permeability to sodium at rest is low. So it's closer to the equilibrium for potassium, equilibrium potential for potassium, but it's not equal to it. Any voltage that is above rest we call depolarizing. A depolarized voltage. Any voltage that is more negative than rest we call hyperpolarized. More polarized, hyperpolarized. When we get to action potentials, after the breakout, we're going to talk about repolarization, because that's what happens after an action potential. Just hold on for now. So the next slide are some clickers. I don't think I printed them out for you, but they're on Moodle. So what I want you to do now is go break out. I am going to open the breakout room right now, reopen it. And then when I see that you're all here, there on the Zoom, I will randomly break you out into rooms. Probably 4 students per room, plus or minus 1. Pull up Moodle so that you can pull up the calculator and pull up the problem set and teach each other, help each other do the problems. And you can focus on the clickers, but I think it's most efficient if you focus on the problem set problems. Okay, so I will reopen the Zoom, reopen the breakout. Remember, you have to go to the breakout Zoom, not the lecture Zoom. Go to the breakout Zoom, and we'll open it up in 10 minutes.